So thanks everyone for attending the ISMT Global event. My name is Mate Path, and it's my great pleasure to introduce Veronique Kiermer. So Veronique is the Chief Scientific Officer at PLOS, where she oversees the editorial department and the development of services, products, and policies to promote open science. Before joining PLOS, she was Executive Editor and Director of Author and Reviewer Services for the Nature journals. And she was a founding editor, Chief Editor of Nature Methods, and holds a PhD from the Université Libre de Bruxelles, Belgium. She currently serves on the board of directors of Keystone Symposia and ORCID. Veronique will be speaking in this general session about a new framework for transparent reporting in the life sciences. There will be time for questions afterwards, so please drop them in the Q&A during the talk. So with that, Veronique, please take it away. I'll stop sharing my screen.
Thank you, Mate. So let me share my screen and put the presentation in presenter view. I hope that you can all see my slides now. So thanks, Mate, for the introduction and for the invitation. It's really
Veronique, I don't see your slides somehow.
You don't see my slides, okay. Let's see. Let's try this again. Are we okay now?
Yeah, now it's perfect.
Perfect. Okay, thank you very much, and sorry about that. Thanks, Mate, for the introduction and for the invitation. It's a great pleasure to be here. I will start with a few disclosures of potential competing interests so that you can put my comments in perspective. And I also would like to have a caveat, is that I'm going to talk to you about the MDAR framework, which was designed for the life sciences. So for those of you who are publishing essentially in other disciplines, I apologize that not all of it might resonate, but I hope that nevertheless there will be commonalities and opportunities for learning and for translation and cross-fertilization between the different fields. So hopefully you've had a chance to absorb the competing interests. Let's talk a little bit about why should we increase reporting transparency. Obviously, we could talk about that for hours, and I have a strong suspicion that I'm actually preaching to the choir in this audience. So I don't want to say too much about this, but I would like to use two examples to ground this presentation in something that I see as a very important need for improvement. The first example is taken from the work of Malcolm McLeod, who is one of the developers of MDAR. And what you see on your screen here is a meta-analysis of several publications where the same drug was tested in the same animal experimental model. And what you see is that the studies that actually reported using methodological safeguards against bias, such as randomization and blinding, showed a much lower drug efficacy than the studies who did not report these measures. So the outcome was 44% less if these measures were actually reported. And that was important because if only the studies that had included these measures to prevent bias had been used as the basis for deciding whether this drug would go in clinical trials or not, it's likely that it would not have gone into clinical trials, but it did go into clinical trials. It went through a big safety trial, and then it went to an efficacy trial with more than 3,000 participants, and it was a dramatic failure. There was no effect of the drug whatsoever. And so that's an example why it's so important to not only to capture these critical elements of experimental design in the reporting of these preclinical studies, so that the body of evidence that is used to make decisions about going into the clinic can be informed by the quality of the experimental design. And despite that being so important, 7 years later, the same group did an analysis of a random sample of papers in the life sciences that used in vivo experiments. And as you see, the reporting, the frequency, the prevalence of reporting of randomization and blinding is actually still very, very low. Randomization has increased over time to reach about 30% of the papers studied, but blinding is still less than, it's still about at 5%, which is problematic. Here's another example. Let's have some facts about cell lines. More than 400 cell lines are known to be misidentified. That means really there is no authentic stock available, and what's in use is actually another cell line that has contaminated the original stock. There is a study showing that for a certain type of cancer, an estimate of 15% of the cell lines that were in use were either misidentified or contaminated. And another study showed that only 5 or 10% of studies that use cell culture have used cells that were contaminated by mycoplasma. Yet, less than 50% of cell lines are unambiguously identified in the literature, and that's the results that you see here from a study by Walizewski et al. In Pure J that really reports, it depends from the discipline of biology, but overall less than 50% can actually be identified, let alone determine whether they were authenticated or whether they were tested for mycoplasma. So I think it's important that these problems are originate in labs where the best practices are either not known or not always followed, and we know that by the time the results are submitted for publications, there is very little that journals can do to quote unquote fix the problem. But I strongly believe that we as journal editors and publishers have an important responsibility in making sure that the reporting is sufficiently comprehensive and sufficiently precise that the quality of and the robustness of the findings can be evaluated by those who are going to read and reuse this information in their research. And I think we can do better. And in fairness, I think that a lot of journals and have tried to do better, and also sort of researchers, organizations have tried to do better. And this is how we've seen the development of a number of reporting guidelines and reporting checklists. And for example, a lot of that was informed by a long-standing effort in the clinical research. I think we have to realize that clinical research has been looking at these issues for much longer than the life sciences, and it's a bit humbling because it also tells us that this is not an overnight pursuit. But I think that we've learned a lot of very useful lessons, and there is I would refer you to the Equator Network that has a number of very well developed reporting guidelines, and for example, the consort guidelines, which was sort of one of the first of its kind, which really has shown to improve the reporting of randomized clinical trials in that case. So that was very much an inspiration for, I think, a lot of life science journals and groups of researchers to develop guidelines specific to the life sciences, and that started in the early 2000s, and it's really accelerated since about 2010-13. And what we've seen, and as someone who's been involved in trying to implement some of these guidelines, I have some observations to share. I think this is going to be very, very summarized, and I would refer you to the two studies that are referred at the bottom of the slide there, that are actually quite informative. I think that the observations we have is really that a checklist on its own is really not enough. There is really a need for follow through and more information in order to be able to really change behavior. The other caveat that we have is that with this sort of accumulation of many discipline-specific and journal-specific checklists, that can lead to author confusion and to an overwhelming burden, right? And a biological paper is very often composed of multiple experiments with different technologies. Each of them could have its own sort of reporting checklist, and you can, very easily get overwhelmed by the number of checklists that you would have to fill out if you really wanted to apply by all these principles. But I think that also, very importantly, the checklists have been shown that they can work. We've seen that from the clinical research, and we've seen that with some of the checklists that have been used and studied through meta-analysis after the fact. And so I think that in particular, if they're short and if they are followed through and really focused, they can have a very big impact. And so these observations were really the motivations for the development of MDAR. So MDAR stands for Material Design Analysis and Reporting. And what it aims to be, really, it's a minimal generalist framework. It's kind of an umbrella for all of these sort of specialist guidelines. It's been designed to be applicable to the broadest possible range of life science publications. It's very important to remember that it complements, but it does not replace and it does not supersede any of these specialist guidelines. Where they exist, the specialist guidelines are still the better guidance for specialist communities. But what the MDAR framework is trying to do is strike a balance between this desire for comprehensive reporting and the practical implementation of that for authors as well as for journals. And also the point to keep in mind throughout this is that MDAR is a starting point, and there is it's very much intended as a starting point with a trajectory for improvement over time. So let me just start by crediting the people who participated in the development of MDAR. The working group, as you can see, was composed of people who have had experience with intervention at the level of editorial policies. There were editors from various publishers, but also researchers like Malcolm McLeod, whose work I've already mentioned, as well as David Meller at the Center for Open Science, who had been very much involved in the top guidelines, which you may also be familiar with. Something to stress about MDAR is that it was very collaborative and it was a very consultative process as well. And so I want to acknowledge here the experts who have allowed us for their name to be mentioned, who have provided very helpful feedback on the guidelines. And I also want to thank all the staff who participated from the journals who participated in the pilot, which I'll come back to a little bit later. So the MDAR framework is actually, it has three elements, three artifacts, if you want. The main one is the MDAR framework itself. And so for each of these rubrics of material, design, analysis, and reporting, it has a set of elements which have minimal reporting requirements, as well as best practice, which is more of an aspiration, if you want. And so this is where you can start to see the evolution or the trajectory for evolution from minimal requirement to aspirational best practice. Accompanying this framework, there is a checklist. Now, I want to stress that the checklist is an optional tool. Journals who decide to use the MDAR framework do not have to use the checklist, but those who want to might find it helpful to have something that they can use sort of off the shelf. And the checklist is formulated to correspond to the minimal reporting requirements of the MDAR framework. And then there is an elaboration document, and that, if you want, is a user guide. It includes more context and rationale for some of the decisions that were made, and it sort of documents the evolution of the creation of the MDAR framework. All of this material is available as companion material to the PNAS publication from April this year, MacLeod et al. And it's also all available on the OSF framework, including additional material about the pilot and so on. So here is a brief timeline of how it was developed. The working group came together in 2017. And basically looked at as input a number of guidelines. The top guidelines, which were sort of journal level reporting guidelines, as well as other reporting guidelines like the ARRIVE guidelines or the Miami guidelines and etc. We also looked at journal policies and sort of, tools, guidelines and checklists that journals might have sort of in the back end that their staff use. And we also looked at meta research about these interventions and what had worked and not worked in the past. And we were also inspired by a number of consensus reports from the US national academies around research integrity and reproducibility and reporting. Based on all of that, the working group came up with this, with a draft of the MDAR framework. And that is, if you wish, an iteration based on our collective experience of what had worked and not, and what was most important and most likely to work in the future. One thing that we all had learned is that it was very important to test things in real life, and that's exactly what we did. We recruited 13 journals who agreed to participate in a pilot, and I'll come to the results of the pilot in a minute. We presented the results of this pilot at a workshop of the National Academies where we solicited feedback from the participants. And then we also did a round of consultation where we sought feedback from 42 experts who had experience with reproducibility, establishing checklists, and things like that. And so based on all of that feedback, we iterated the draft and provided a new version of the framework, the checklist, and the elaboration documents, which are now available with the PNAS publication. So just giving you a sense of the pilot we ran, we tested the checklist as a proxy of the framework. And the primary outcome of the pilot was to determine whether the framework and checklists were articulated with sufficient clarity that a manuscript could be evaluated against it and determine whether it met the criteria or not. And then the secondary outcome was to get a sense of how both authors and editors, how they were feeling about the usefulness of this material. And so for this secondary outcome, we ran a survey which had more than 200 responses from the corresponding authors of the manuscripts tested and 33 journal staff. And what you see here on the slide is the results of that survey. And overwhelmingly, more than 80% of both editors and authors felt that the checklist was very helpful or somewhat helpful to make sure that the manuscript complied with the reporting requirements. The participants on the side of the journal staff kept time, and overall we estimate an average of 24 minutes per manuscript or 15 minutes median to check the entire checklist against a manuscript. And then 89 of these 289 manuscripts that were evaluated twice by two journal staffs who were blinded to each other. And then we compared inter-assessor agreement, and we took low agreement to indicate that there was not enough clarity in how the questions were formulated, and that also led to improving and refining the descriptions in the framework and the checklist. So that's how the MDAR framework was developed. I hope it gives you a little bit of comfort about the fact that it was actually tested in real life. The other thing that I want to stress is that, it's really, MDAR is designed to be modular. So for the journals who want to use MDAR, it is absolutely possible to adapt it to the needs of that research community. And I'll give you a couple of examples. So the first one is really that at the level of the framework itself, there are two things. There are the minimum requirements for each element, as well as best practices. And we recognize that some communities might be further ahead with some elements but not with all elements. And therefore, a journal can decide which of the minimum requirement or best practice they want to use. Similarly, by extension, the checklist is modular. As I said, it's completely optional. You can use it any way you want. It can be author-facing, it can be backstage. The minimal requirements and best practices can be swapped, and you can also organize it in a nested way if some rubrics are more important for your community than others. And then finally, there is some flexibility at the level of endorsement. So let's talk a little bit about endorsement and what it means for a journal to endorse MDAR. It means that the journal expresses support for the transparent reporting principles that are outlined in the MDAR framework, and that the journal commits to taking steps to incorporate these guidelines into journal practice. So it's really sort of a commitment towards improving and going along the lines of MDAR. Having endorsed the framework, journals can decide to implement it at 3 different levels. The first level is recommend, which means that the journal endorses the framework and recommends that authors consider relevant aspects of MDAR. So this is quite minimal. It can be a paragraph in author's instructions pointing people to MDAR and suggesting that they take that into consideration. The next tier of implementation is the limited mandate, by which a journal endorses, recommends, and requires compliance with a limited aspect of MDAR. And then the last tier, the more stringent tier, is the full mandate, in which a journal endorses and requires compliance with all aspects of MDAR. I just want to say that endorsement is also important because it can help stimulate interest from other stakeholders. It's clear that this is not, these issues around reporting and it are very journal specific, but they are the symptoms of best practices and things and culture change that needs to happen in the research ecosystem as a whole. And therefore it's going to take more than journals. And I think it's very important to try to engage other stakeholders, and the MDAR framework and these guidelines are one way of doing this. We've seen, from previous experience that researchers, for example, use one of these checklists to recommend for new members of their labs to actually consider that as they design experiment, because at some point they will be asked to report on it. And that's very important, sort of like trying to front load the awareness of these elements of experimental design and reagents and so on. Similarly, it's quite important to try to align with the funder requirements and the journal requirements, so that we can actually bookend the process with the same expectations for authors, and really to try to make it easier for authors to know what's going to be expected and to pay attention to that very early in the process. And finally, I want to stress that we've been very pleased to see that there was attention also from tool developers, and I'll mention SciScore here, who have started to use natural language processing techniques to try to extract the answers to the MDAR checklist from manuscripts. And this could be, when it's fully developed, could really be a useful tool for implementation in the future. What's very important to remember is that MDAR is meant as a community resource. It is stewarded by the Center for Open Science on their website. It doesn't belong to anyone. It's really meant to be used by all the stakeholders, but also to be improved by all the stakeholders. And that's going to be very important to continue going forward. Here is one example of MDAR already in implementation. Valda Vincent from Science was on our working group, and she's really led the charge. All science journals now require the authors to fill out an MDAR checklist in the life sciences and publish it alongside the manuscript. You see it here in supplementary material. They have a limited mandate, and they're actually quite close to a full mandate. So I will call out, finish with a call for action. I really hope that you will consider endorsing MDAR and implementing it at the level that is right for your journal community. I think that community action will be very important to make it easier for authors in the end and to really help facilitate this culture change. I will stop there and take questions. My email is there if you have questions following this talk. And I also want to call out the emails of the two corresponding authors of the PNAS publication, Malcolm MacLeod and David Meller. And I'll be happy to take questions.
Thanks a lot, Veronique, for this really nice talk. It seems like a lot of work putting together this MDAR framework. So until we get some more questions in the Q&A, maybe I can start with one. So you mentioned that it's a minimalistic framework. So I was wondering how you decided on what reporting is not absolutely necessary. So maybe in other words, what should go in the best practice recommendation versus the minimal requirement. You can elaborate on this thought process a bit.
Yeah, I mean, it is a subjective process, but it was based on experience and very much based on our sense of community readiness for certain things. An example of that is the example I showed in the slide is the cell line authentication, for example. I think that based on studies that have been done in some of the journals, it was clear that the community is not yet implementing that practice. And frankly, if you ask for that at the very end of the process, it's not it's probably not worth it at the level. It's really something that needs to be done at the beginning of the process. And so based on what we knew of community adoption, we felt that most communities were probably not quite ready to be able to report on that, but that it was important to sort of raise awareness. So we decided in that case that unambiguously identifying the cell lines was the first thing, and that the best practice was really to go towards authentication. And we hope that in the years to come, we would be able to go there. But so it was very much based on experience and community readiness.
Yeah, thanks a lot. So Melanie Slavic has a question. If you have a recommendation about whether to require or recommend use of MDAR at original submission or at revision?
That's a very good question, and that's very much for journals to determine what's best for them. In my personal experience, having tried to implement some of these sort of checklist requirements, I think it very much depends on the selectivity of your journal, and if you have a lot of a very high rejection rate, it is actually. It's at the level of revision that authors are really motivated, because they know that they have a really good chance of, since they are being offered the chance to revise, they have a very good chance to be able to get published, and at that point they are very much more motivated. The problem if you do it there is that it's less helpful for reviewers, who if you do it at submission, it's something that reviewers can have a look at. So practically, in my experience, what has been done is for journals which were really highly selective, did it at revision, and journals that were less selective, where that was less of a motivation, did it at submission, so that it was an additional tool for reviewers who could actually have a look at the checklist and compare.
Yeah. Thanks a lot, Veronique. So in your slides, you also mentioned that checklists can work if followed through. So I was wondering if you have recommendations of how journals can efficiently make them or follow them through. Like one potential issue, I would think, if with a broad checklist where authors have the chance to put a not applicable, what if they put an NA to something that's actually absolutely critical for reproducibility of the work? So if you have any thoughts on this.
Yes, I mean, I think that that's a very big, again, it kind of also depends on the resources that journals have. Journals that are well-staffed. So one thing we've seen, for example, is that can work is to force some of the reporting at the copy editing stage. For in vivo, I know that the Nature journals, for example, at in vivo experiments must report whether it's randomized or blinding, and basically there the copy editor will add it's not being randomized if it was not mentioned, and that's the stage where you can do that. So that's one way, but that presupposes that there is sufficient resources to do that. I think that the first step there is really transparency, and I think that if you can follow through and have staff say, no, this is really important and you need to, and it is relevant, that is a way of really doing that follow through. If you don't have these resources, just actually even what science is doing, which is publishing that checklist, readers who are specialists in the field can actually tell whether it's important or not, and if they say that randomization was deemed not applicable, but it was, that will be helpful to make a judgment, an informed judgment on that. So I would say transparency is the first step. If you have the resources to then follow through and make sure that everything that is applicable is actually reported, that's the best. But we have to realize that that's not the case for all journals.
Yeah, thanks a lot. So since we're now out of time, I just want to thank again everyone, and thanks Veronique for this lovely presentation and the discussion. And just a reminder to everyone to use our hashtag ISMTGlobal to find and share content, and please make sure to check out the next session on innovations and best practices from the era of COVID-19, and to continue this discussion also in the exchange forums on reproducibility or email Veronique if you have any follow-up questions. So yeah, thanks a lot, and see you around in the air meet.
Thank you very much. See you around in the session.